As we kick off the fall 2025 semester at NC State, it's important to remember that research happens around the year. Today, one of our researchers is here with us to share how she uses the herbarium in her research.
Hello, my name is Kira Lindelof. I'm a doctoral candidate working with Alexander Krings in the Department of Plant and Microbial Biology. My research broadly focuses on the conservation biology within the genus Houstonia in the coffee family, Rubiaceae. And more specifically, I incorporate ecology and biology to address questions about things like ecological niche, population genetics, seed biology, and taxonomy in the endangered roan mountain bluet, Houstonia montana, and its congener, Houstonia purpurea. I utilize the herbarium in a few ways, one of those being to house voucher specimens that are associated with my research. A voucher specimen is a physical record that ties my research to an individual or population from which I sampled from. And the specimen is often a whole plant, but can be fragments of a specimen as well, so things like plant tissue. These vouchers are then deposited at herbaria, where other researchers can access them to review my work or to incorporate into their research as well. I incorporate voucher specimens from other researchers into my own work. One of my projects aims to look further into the morphological differences between Houstonia montana and Houstonia purpurea. It's likely that these two species hybridized in the past, followed by generations of hybrids breeding with Houstonia purpurea. Consequently, there are a few instances of what I call morphological intermediates, where those two species distributions overlap, which makes it really difficult to identify those intermediates to a species, and this can be problematic when monitoring populations of the endangered Houstonia montana. One of my goals is to morphologically compare Houstonia montana and Houstonia purpurea to see if we can identify characters that distinguish the two beyond what we currently use. I then hope to apply those to the morphological intermediates to see whether we can identify those as one of the two species, or at the very least relate back to the genetics to better understand the gene flow across populations and species. So when working with endangered plant species, I'm limited to the number of voucher specimens that I can collect. Therefore, I rely on voucher specimens of Houstonia montana that are housed in the herbarium. I pull specimens from the NC State Herbarium and from other institutions like the Herbarium at Chapel Hill or at the Smithsonian Institution to capture that morphological variation that I otherwise couldn't grasp from things like photos on the internet. And it's with that I can really identify those small characters that may be utilized in this work.